This is the first one. The first one.
Yeah.
And they move. Okay.
What's the is it an air purifier or something?
I think it might be air purifier. It could actually be a heater.
Oh, okay.
They heat the glass houses before it provides heating for this building, you see. It's pretty impressive. It is impressive.
Well, I mean, I'm happy.
And the building itself is happy, but it wants some sort of award anyhow. So, yes,
right. Yeah, that makes sense.
Just we're going to start in 2 minutes. Just talking as well to the people remotely. And you have it in that.
I can copy onto here.
Okay.
It's the easiest.
I think that'll be easier. So we
move this out of the way.
Yeah.
Oh, no, that's not bad. The face will be on board of the screen. I'm always amazed it works really well.
Yeah.
Actually, you can browse the internet and that, because if that's the case, but you're connected, and you think that you can browse it, just to check, because if that's the case, what I will do, let me send you the link to the sample, and then what you can do is you can share the screen. But can you check that this working? Because can you check with anything like the browser, anything that is working?
Oh yeah, the browser.
You've been connected. Okay.
Yeah.
Now let me do the following thing. If I send you now the link,
okay.
When it's your turn, you can just share your. You can log into the Zoom call.
Okay.
Share your screen, and you can do it from your computer. Yeah. Okay. Let's do that. Because it's going to be easy. Okay. I send you the link in a minute to your email. Your email. I think I have your email address because you sent it. Yeah. Let me do that.
And I've got it on there.
Yeah, but I think it's easier. You do it from your computer. Fantastic.
Okay. I have a plan B.
We have a plan B.
I'm going to be in the way. I'm just going to keep pressing my little thing.
Okay. Welcome back, everyone, to the afternoon session of this really nice day. We will start the afternoon with Haidee's presentation. Haidee's been leading the work we do in statistics here at Niab for many years. Doesn't matter how many years, but you had the chance, of course, to work with Mac over those years, and fantastic to have you here today. Yes, hand over to you. Thank you.
Thank you. Okay, well, welcome everyone. I'm going to take you back in time a little bit here, and I'm going to talk about a paper that I worked with Ian on, looking at the contribution of genetics to the improvement in crops over the time. And I realize I don't know which one I'm using for improvement slides on them, presumably. Not that one. Oh, just that one. I'll just do that one. Okay, thank you. So, what started it for? Well, it was Wayne, really. Although thinking from Phil's talk this morning, I'm wondering whether it was actually Ian rather than Wayne. But I do remember Wayne coming to see me and basically asking the question, what are breeders doing? I slightly paraphrase, it wasn't quite that word. And talk of the time was, are all the varieties the same genetically? And I'm never quite sure whether we were trying to answer the question, what are breeders doing? Or whether we were trying to answer the question, to prove that breeders were doing something. But we tried to start to look at the genetics within the historical variety sets that we had. And we did this using published data, which is quite limited. You get a year mean per variety over a number of years, but that's all you get. And BSBB quite quickly got involved with the discussions on this, and they and the Niab Plus funded a project where we could use a trial mean data set. So we had more data, we had more precise data for locations. And after probably 18 months or so, we actually presented this data, and this is basically what this talk is about. And, oh, sorry, wrong one. This is the first slide of the presentation that we gave to BSBB with our results. We were building on work that had been previously done by Valerie Silvey, and she had looked at the contribution of varieties to the overall yield, taking data from 1947 up to 1978. And we built on this pretty much. And this was the summary that we gave. I'm not intending to read it all, don't panic. We were incredibly pleased that when we repeated Sylvie's work, we actually had very similar results. And we were pretty amazed, really, not least because everything pre-78, we had actually electronically inputted from hard copy that had been found in various cupboards across Niab. We were also slightly annoyed to realize that Sylvie had stopped at a very opportune time because the trialing system changed after that, which actually caused us all sorts of problems. So what did we do? We actually analysed the data from 1948 up to 2007. And I think as I go through, it's quite important to remember I stop at 2007 and look at what proportion of the yield increase is actually genetic. And we did this for a number of crops. We have a huge data set. The early years were pretty much only heavy yield. The latterly, and certainly in the 90s, there are a number of characteristics. That was the time of a lot of data. It's since compacted a little as to what's now recorded. We have less years for the oilseed rape, the maize, and in this study for sugar beet actually is the longest crop that we have got recommended lists for. They actually go back to the late 1920s, but not electronically. And we only looked at the electronic stuff. So you can see there was plenty of data. And we, as I said, we finished in 2007. We also used varieties that had a minimum of 3 years of data, partly because as it goes through the statutory process, a number of varieties will be dropped after 1 year. A further number will be dropped after 2 years. And so we just determined that 3 years was really the minimum to get some linkage across what is actually a 60-year time span. And this is what we found. I'm not going to do any great detail in this. Suffice to say the yellow dots are the varieties in each year. And the line is the estimate of the yield effect. Well, actually, the big line is the estimate of the yield effect, the year effect even, having been adjusted for the varieties. So we did this for wheat. What was interesting is this if I show this one, if I take out that line, you can see the genetic variance for wheat. And one of the questions was actually fairly simply answered, that we seem to have the same variance genetically in the latter years as we did in the early years, which was quite reassuring and very nice. We actually had very similar results for winter barley. I'm not sure if I can show you on this, but if you look at 1981, you can see a large upward movement of the year effect there in the blue line. And that was when the trial management switched over from untreated to treated management. So fungicide was being applied after that, and that had a huge effect on the year. Had very similar results for winter barley. Spring barley followed a similar path, but there was less of a year effect differential. Okay, for sugar beet, what we found was actually the year effect is increasing as well as the variety effect. So basically, agronomic practice is improving at the same time as the genetic improvement that you see. Ian was very keen to point out that some of this was actually genetic, even though it's put as agronomic. And he always cited sugar beet bolters, where the breeders have made great efforts to develop varieties that didn't bolt, they didn't go into flower and therefore be worthless, which allowed you to have a longer growing season and therefore more yield. And so even though it's in the agronomic part of our analysis, it was actually a genetic improvement. And Ian was very keen to point that out to whoever was around to hear it. Forage maize, we see something similar, not unsurprisingly, because it's a much newer crop to the UK. We're still seeing the agronomic improvement. We also looked at some quality characters, so it wasn't just yield. And we looked at the oil content in winter oilseed rape and the sugar content, which is actually the important bit of sugar in sugar beet. And these, again, are showing the same sort of thing. One of the tasks that we were asked to look at is how our trial results related to the national yield. And so we spent quite a long time trying to figure out how best to match this up. And you can see here, just for wheat, that there is a very good correlation really between there was a very good correlation between the national yield and our treated trial data. And so we were able to plot the effect of the variety improvement on the national yield. And maybe, strange, I thought I hadn't the slide, but never mind. And this is particularly prevalent since 1982, actually. This actually came from the paper, and this looks at where we estimated the contribution of the variety to national yield increases. You can read for yourself, the blue diamonds are the national yield. The green line actually is a spline, but that's been fitted to remove the fluctuations. National yields year on year do vary quite dramatically. And the red line is the contribution of the variety effect, or what's down to the genetics of the variety. So you can see the genetics and the improvements that the breeders have made are pretty impressive in their own right, and definitely fundamental to the improving yields. Always with an eye for future projects and funding, Ian then started to look at other things, and he was very keen on disease resistance breakdown. And we looked at how the longer a variety had been in trial, the more likely it would be that it broke down to disease pressure. And it's clear that there's a very good correlation with this. And this, I should say, is actually just on average, but obviously each variety has its own makeup. And if we separated these out, we can see that moving to the right of the screen, Brigadier and Schnettner were two known varieties that broke down to disease pressure. What's interesting here is the variety in the middle of the two is actually Oakley, which broke down very dramatically in 2009. And here we were with 2007 data predicting that it was going to be vulnerable. And we both felt that that was quite an important point, and we both had hoped that we would get further funding to look at this more closely, but it never materialized. We also looked very briefly at climate and what influenced the varieties. Dominant was summer rainfall, which maybe is obvious, but equally dominantly was winter temperature. And this is wheat, so that was interesting. Cold winters was definitely one. I can't remember what I put in the slides. So this is cadenza, and this was very susceptible to the summer rain. Cadenza is actually an autumn sown spring wheat, so that's perhaps not surprising. That will be in its genetics to be like that. Malacca was very susceptible to the winter temperature, but there is a lot of noise in this data. Again, we had hoped for some future funding to look at this more closely. It didn't happen at the time, but the last thing I worked with Ian on was to support a PhD student, Joanna Raymond, and she, not long, I can't remember which year, but not long ago, finished her PhD, where she had not only updated our analysis, but she had also introduced climatic modeling into her thesis. So that's very well worth a read. And she's a very good PhD student, and she's done some very nice work. Okay, so this is when I'm going to start to struggle. Ian was always incredibly good at making me think I understood all of his genetic stuff until I left the room and I realized I hadn't really understood any of it. So this is much more Ian's work. And we started to look at the genetics of the variety. Everything else had been sort of stuff we found in fields, basically, observations from the fields. But this was the genetics. And this was associative mapping, which was very in vogue at the time. And here we've managed to identify winter and spring barley and 2 and 6 row. So a pretty picture, but pretty worthless if you're actually trying to study anything, because those are known. And this was just looking at the marker differences. The most I ever got from this is everything on the left was good and the things on the right were going to really muck your head around. And there was too much on the right to be useful. But it does work when you're looking for a simple trait. So, and that's my only picture. So I'm determined to keep it in. And this is the gene, I guess. So it's clearly very, very good for that. Ian's great love was genomic prediction. And he was very keen to move into this. And so back in 2007, we did some, or he did some. I didn't really understand it. He really felt that this was the future of plant breeding, which at the time wasn't quite so common. And we looked at, I don't know, I'm sure everyone knows this other than me, but you have a test set. You basically divide your data in half. You have a test set and you try and predict your other half of data from a training set, and you try and predict the test set. Is that the right way around? Yes. And you can see we actually get an incredible correlation if we just divide the data in half. This was the best slide, and this is the one that Ian showed. If we actually used old varieties to try and predict the future, it was disastrous. However, we did, according to Ian, get acceptable correlation if we didn't separate by more than 10 years of data. I had to trust his luck to that because statutory authorities would never allow this to be acceptable. And then I am literally going to finish. Basically, this was our conclusion. Ian was a firm believer that the genetics should play a part in the overall variety performance results, and we should be using the genetics in those results. And then just a wee bit of, there were many people involved with this, but it's no secret, no doubt, that overall it was Ian that drove it forward. And I think, if I'm honest, it probably took me about 3 years after we gave our presentation, and certainly after the paper, before I even realized that this was actually considered quite important. I just thought it was one of those things that you just did and forgot about. So that's it. Thank you.
Haidee, no, thank you very much. And before you go, just my comment is that you are really working at the cold phase of looking into the varieties and how these are coming through the pipeline, which I think is something which I remember Mac enjoying a lot. I think that kind of interface. But one point you made, which I thought was very interesting, this idea of being able to be predicting, if you like, the future. And there are now different ways we probably will assess varieties. And I wonder how much of this you think is more of the same, or is going to change the way you work?
I think it will be fast and furious. It will change the way we work. I think people are targeting specific genes, traits. They can see an end use for it. To me, from what I've learned, knowing your end use and being able to show that it's got a market value opens up all sorts of doors through that testing process. Yeah.
Thank you. Any question, any comment? How many cupboards and drawers we have in Niab? Far too many, probably, right? You can find it.
At the time, there were quite a lot. Yeah, finding the keys for some of them proved quite a challenge.
It's a data problem at times, right?
Yes.
Thank you, Haidee. Good. The next speaker is Tally, and Tally, I think probably you will say more, but here you've been mentored by working with Ian, and you have been in many ways taking this post to the next stages, and you will be presenting something what you're doing. And let me make sure you stop sharing here.
Oh, thank you.
You can share.
Okay, thanks, Mario. Yeah, hi everyone. Today I'm going to be talking about a piece of work that's been going on for several years at Niab, and this is focusing on accessing genetic diversity from wild relatives and trying to incorporate that into the modern gene pool of wheat to improve UK wheat sort of elite breeding diversity. So this work here was probably Andy Greenland's brainchild originally, although I'm sure Mac was involved over the years. In more recent years, he's been helping me, as Mario said, as a sort of an advisory role, and he's been a wealth of really useful information, and all the analysis you'll see in here I have been discussing with him, and he was helping. So he's been a real big help to this work. So I'll be talking about some of Niab's wheat diversity resources with a particular focus on synthetic hexaploid wheat, how we've been exploring this diversity through genetic mapping, and also looking at genomic selection as well, which is what we're thinking about moving into now for this piece of work. The challenges of working with this material, so some of my thoughts, and how we're taking steps to overcome some of those challenges going forward. So this is a room full of people. I probably don't need to tell that multi-founder populations are powerful for genetic resources. With a focus on nested association mapping panels in particular, these are really good for having over, say, 50 different donors and capturing that information into a closed population. We do that by crossing those 50 different donors with a common parent, in our case, back crossing into a UK adapted wheat background. So that's what we've been doing. We've made 4 really large nested association mapping panels as part of the Designing Future Wheat programme. And this has been a collaborative effort with loads of people involved, but Richard Horsnell was responsible for the synthetic NAMs and Fiona Lee was responsible for the tetraploid NAMs. So I've been working on the winter wheat synthetic hexaploid NAM. This is where we took 54 synthetic hexaploid wheats and backcrossed them into an elite background, a winter wheat background, which is Robogus. And that was over 3,000 individuals in that population. On the spring side, it's even larger, and that's backcrossed into an elite background of Paragon. We've been doing this with tetraploids as well to access the genetic reserves of the AB genomes, with the synthetics focused on the D genomes. We're also looking at the tetraploids Durum, cultivated Emma, and wild Emma. I won't be focusing on the tetraploid NAMs as much today, but just to say that the resource is there. So this work has just been accepted for publication in TAG, and I've got to submit the final edits tomorrow morning. But this is the kind of overview of how we created the primary synthetics. So for those of you who don't know what the primary synthetics are, it's a way of going back to the hybridization event that happened 10,000 years ago in the wild, where a tetraploid hybridized with a diploid goat grass called Aegilops tauschayi. So what we do is we resynthesize that in the lab with a tetraploid donor, typically a Triticum durum line, synthesize it with lots of different Aegilops tauschayi lines to access the reserves of the genome donor. That forms a kind of unstable triploid, which you then use culture seed to get into a primary synthetic, which is a hexaploid. At this stage, it's really diverse, but it looks like crap. So it's not really useful for a direct breeding application. So what we then do is backcross it into a UK winter wheat background to make it more of a sort of oven-ready source for pre-breeders. And this touches on what Sarah was talking about earlier. So we did that with 54 primary synthetics, and then made lots of different nested biparental populations from each of those crosses. And that's what was taken through SSD in the glasshouse to go out into the field in what we called the core nurseries. So due to the population size, we had the core nurseries over 5 years. And in those nurseries, we assessed for traits such as flowering time, yellow rust resistance, and plant height. Breeders would also come to the core nurseries and make selections. So as part of the Designing Future Wheat programme, which is this large project that this work was involved in, the breeders would come and make selections on what lines to incorporate into their own pre-breeding programmes from this large nested association mapping panel. So if we look at some of the genetic makeup of the population in the PCOAs here, so in that top middle one, you can see Robogus in the far top right-hand corner, and then we have the individuals in the green clustering slightly closer to Robogus, which we'd expect for a backcross population. Then we have our primary synthetics there as well. In the plot below, we're looking at just a D genome of the primary synthetics. So you can see that blue cluster there with the dark blue line in, that's Robogus, and that's the lineage 2 Aegilops tauschayi cluster. So that's where the D genome donor of bread wheat came from. In the far right-hand side, we have the kind of yellowy orange cluster. That's lineage 1, so that's meant to be the kind of untapped lineage of the species that haven't really been utilized in modern breeding. And then we have this kind of intermediate dot, which is one Aegilops tauschayi that's in a primary synthetic that's meant to be this new lineage that's recently been discovered from Georgia. So as you can see, the D genome component of our primary synthetic sampled the lineages of these Aegilops tauschies, goat grass species, and across their geographical range as well. So yeah, every nested population within the NAM, which was 54 of, was made up from a different synthetic donor. And across those donors, we had 47 unique tauschies and 15 different tetraploid donors as well. So we did experiment a little bit with tetraploid donors. We didn't create all of the primary synthetics that we used in the study. Some were from the CIMMYT winter wheat program and also some from a university in Romania. So 5 years of core nurseries where this material was grown out into the field as it staggered in its development. As you can imagine, phenotyping this was a bit of a nightmare of over 3,000 individuals. So in 2021, we took all our leftover seed and put it into what we called the Mega NAM nursery. So this was at the time Niab's largest ever winter wheat nursery. Later in the year, the next year, we've beaten that by growing the spring NAM, but at the time this was big, and you can see the size of this of the video here. So the Niab Data Science Department flew drones over as well to collect some more informed trait data. The trait data that we focused on for the paper is flowering time, plant height, and yellow rust resistance, and that's to try and characterize the material. To summarize these phenotyping results, the primary synthetics were early flowering, they were taller, and they had better yellow rust resistance. With the that's compared to Robicus, and then the population individuals sat somewhere in between, Robicus being the recurrent bred wheat background. So what was really interesting about this piece of work is that during the core nurseries, breeders from these different companies came and made selections on the material for incorporating that data back into their own programs. And also some of the lines that they selected were then advanced to later stages in the Designing Future Wheat program. So we had several breeding companies visiting, and we can't share the details of what was exactly selected, but you can look at the kind of trends in selection. So where in this case, blue represents a genotype that was selected, and a genotype being a synthetic derivative, a member of the NAM population, was selected by a breeder, by 50% or more of the breeders during the core nurseries. So the breeders tended to select towards better yellow rust resistance, as we'd expect. But then in terms of plant height and flowering time, they tended to select towards the middle, which is probably the Robicus mean. It's also really interesting to see which in the lines that were selected. So in these plots, orange represents a genotype that was selected by any breeder across those core nurseries. So it's interesting to look at the lines that were selected. What is the original tetraploid or Aegilops tauschi lineages in those lines that were selected? And as you can see, most of the Niab synthetics were this Ho 501 background, which is the most commonly used tetraploid background, when actually some of the rarer tetraploid backgrounds had a much higher selection proportion by the breeders throughout the trials. So in some cases, up to 70% of the genotypes put out were taken forward by breeders, suggesting that maybe the Ho 501 background isn't as popular. Lineage 2 was the most used Aegilops tauschi lineage, but and was also the most selected as well. So a lot of the work that I've done for this is on the genotyping side, and phenotyping the population is a bit of a nightmare. Genotyping is also a little bit of a nightmare. We extracted the tissue, or Richard extracted the tissue here at Niab and sent it away to Bristol University to be genotyped from the 35K Axiom array. Got the data back, and to get as much value as we could from the marker data, I went through all the markers manually, inspecting each one to make sure we were retaining as much as possible. That then went through marker quality and individual quality controls, imputed data with random forest, anchored to a physical position, and then produced pruned marker sets for kinship estimation, and then feeding into association mapping pipelines. This data is all now available on our Niab GitHub repository, so this goes alongside the paper that's about to come out. And flowering time, plant height, and yellow rust for the traits we did the QTL mapping on. So I'm showing an example here for the yellow rust data, and this is the yellow rust results from the 3 trials that yellow rust was measured in. And what you can see is that there's a really nice consistent hit on 4D there. And this is a really good example of a beneficial allele that we're capturing from the synthetic background. So here, this hit, the peak hit, same for every trial, blasted 1.4 megabases on 4D. And that's 400 kb away from a known yellow rust gene, which is common in a synthetic background, or had been identified in a synthetic background before. So we think it's this gene. It's really interesting because based on some of the Chinese papers I read, this gene isn't very common in many land races or cultivars, and there's no evidence of gene success in a UK wheat background until what we're showing here, that it's working in a Robicus background. Now, Robicus isn't really a recent variety, so it might be that we need to confirm that in more recent varieties. But even looking at just the primary synthetics themselves in the field, you can see the ones that had the resistance allele had a much, were slightly better in terms of overall yellow rust resistance, same for the population line. And when you also look at the lines that were being selected by breeders, they were significantly enriched for this QTL. So it's obviously was impacting selection. So the other side of the work that I haven't spoken about so far, and why these synthetic hexaploid wheat derivatives are quite a unique pre-breeding resource, is they have really competitive grain yields. In this case, you can see by that red circle there, I've highlighted Robicus in this trial, and there's a lot of synthetic derivatives there that are outperforming their recurrent parent, and even going high enough in yield to be in line with some of the more recent elites. So as a pre-breeding resource, that's quite rare as far as I understand. And it's the fact that they have this competitive grain yield combined with all of these kind of novel introgressions that we've got across the D genome and also the A and B genome as well, makes them really desirable candidates for pre-breeding. However, when you do a GWAS for yield, as we've done here, you don't actually get any QTL results, which is interesting. So this data here, this is where we've teamed up with Karis from KWS. So Karis has really kindly given us the yield data back from the lines that he selected from those core nurseries. And we can use this to do a GWAS. We've got 406 lines here with genotype and phenotype data. So it might be that, as we know, yield is very polygenic. So there's a lot of QTL involved, and we might not necessarily be able to detect them at this stage. Also, because Karis and the other breeders have sampled from lots of different nested populations, we've got a lot of different backgrounds in this data set. So it might be that we don't have the power to identify a decent marker trait association due to that. And that's why we're a little bit more interested in genomic selection at this stage. So here we've got our 406 genotypes with data available. So we can treat that as a training population, partition that between a training population and a cross-validation set to work out how good our prediction is. And when we're happy with that, what we've done is run genomic prediction using GBLUP, and then predicted the test populations. So the test population is, we have 2,231 lines available to predict on that haven't been tested at KWS. So that would be great, but we actually don't have that much seed available for the majority of these lines. And that's because in that mega NAM nursery, due to logistical reasons, we couldn't combine. So everything we wanted to take, we had to hand sickle. So we hand sickled around 900 genotypes, 900 plots, which is a lot of work. And we can make selections available on these for 572. So those 572 came from 15 nested populations that we selected, which have either really diverse donors or breeders had favoured lines from those populations in the past. Oh, and just to say, that's what we've done. We made the predictions, and now what we've said is we will take the 25 best predicted, the 25 worst predicted, and 25 most similar to Robicus, and grow them in 2 independent evaluation trials at KWS to work out how good the prediction is. And that's currently what we're doing at the moment, so it'll be interesting to see how it works. These are the observed values from the training population. So what you can see here is that some nested population, this is organized by nested population, some nested populations are really well represented, some not so much. And that might be that when we bootstrap the genomic selection analysis, as you can see in the top right-hand corner there, so picking a cross-validation set from the training population at 10% and 25% is a little bit unstable. So whatever ends up in your cross-validation set, I think, depending on how much data you have about that in your training population, is how good you are predicting it. So there's a little bit of error there, and it'd be interesting to see how we can improve on this going forward. Even looking at what's predicted in the test population from those 15 families, it's really interesting to look at. So these are, again, organized by nested population. This lower predicted population here, this is the population that came from the lineage-free donor. And it's also in that HO501 background that I was talking about, the breeders not tending to select towards, and also this kind of new lineage. In this case, again, a lineage 2 donor, but a HO501 tetraploid background. And in terms of the other side of the scale, we have the tetraploid background that was selected most by breeders in the core nurseries. This is lineage 1, and a few genotypes from this population were selected by every breeder, which is interesting. And again, this one had nearly 70% selection in terms of the tetraploid background, lineage 2. So based on what the breeders have been selecting and what we're predicting, it makes sense. So hopefully there is going to be some correlation in the results. So just thinking about some last thoughts about the challenges of working with this material. Definitely size, it's really big. Phenotyping, genotyping, there's a lot of work. Adaptability, I think we've spoken about this today. A lot of the crosses that we're making, we're undoing a lot of good work. We end up with quite a lot of rubbish, and it's quite a wasteful process when you think about it in that sort of sense. Pre-breeding value, we have this thing where we try to maximize the amount of pre-breeding value in the material. So took different back cross streams forward in each nested population. That's really great for making lots of diversity available to breeders, but not as good for statistical mapping because you have less representation of that exotic haplotype that you might be trying to make a marker trait association with. Then also marker coverage across the D genome. The array we use a genotype on was based on diversity observed in modern wheat. And this diversity is enriched across the D genome, so we need more D genome markers. So here are some thoughts about how to overcome some of these challenges. Definitely high throughput phenotyping. So we're working closely with the Niab data science group here in our UK-based trials. We've currently got the spring NAM growing in India at ICRISAT in collaboration with ICRISAT and Sigrid. So this is to screen for heat tolerance, and you can see the scale of the experiment here. So this will be repeated twice for the synthetics, which is what you can see here. Then also on the tetraploid spring NAM as well. And also, teaming up with Caris again. So we've just had a PhD studentship funded. I'm interviewing for that post next week. So we're going to have a student who's going to come in and look at trying to optimize diversity capture with pre-breeding simulation, with a particular focus on back cross breeding and trying to retain major adaptation genes in your breeding pool. They're also looking at exploring combining ability of different elites and diverse donors, and exploring haplotypes and how exotic introgressions recombine in elite backgrounds. This idea about pre-breeding value versus statistical power. So this is a linkage block within the NAM that you can see here. And as you can see, there are some well-represented exotic introgressions, but also a lot of less, well-represented exotic haplotypes, but also some quite rare haplotypes. A way around that, and very suited today, is to think more about a magic population. So that's what we've been doing in the background, and this is meant to be kind of the follow-on for Magic Diverse and Magic Elite. It's called Magic Wilder, and what we did was we took the best spring BC1 genotypes in a spring background, and these parents all had introgressions from synthetic wheat, cultivated tetraploids, cultivated emmers, and wild emmers. So a lot of diversity in there. And that's now currently at the final stages of SSD and 1,000 genotypes. We just need to find the money to genotype it now. And then improving marker coverage. So a lot of the Taoshis that I've been speaking about today have actually been sequenced as part of a collaboration between Niab and Branda Wolf's group. So the sequence data is available for the D genome donors in some cases. Our long-term goal is to sequence the NAM populations and also the primary synthetics. Bristol University have just released what they call the next generation SNP genotyping array for wheat, and this is meant to be more based on different diversity pools, have better coverage across different chromosomes, and we've just regenotyped our primary synthetics on that new array, so it'll be interesting to see how good that is. So a lot of different people are involved in this work, which has been a really collaborative effort from the Niab team, and lots of people have been involved. A big thank you to the breeding companies, some of which who are here, and some people here made those selections. And we really appreciate their involvement and Design for Your Wheat project. I also really want to acknowledge Ian's help that he's provided me for the last couple of years since I've been in a more senior research role at Niab. It's been a real pleasure to sit in a lot of consultancy meetings with him when he's helped other scientists as well, and hear how much he cares about their science, how patient he is with people. It's been a real pleasure. And I wanted to include some slides here. These are from the communications team, and these are where we compiled the photos from as many different years as we could from Ian's quantitative methods in plant breeding course. The earliest one we can find was 2009. So this is the old teaching lab, which was a really unique building a unique room in the old building, which is well suited because Ian's course was also very unique, and it was something that was really special. And you can see a lot of very familiar faces in these pictures, and I think that's a testament to how many people he's helped over the years. So yeah, that's everything I wanted to say. Thank you.
Thank you. Well, thank you, Tally, and thank you for finishing with this. It really is a massive part of the legacy that we have from Ian in terms of the impact beyond Niab, right? This course. But do you have any questions for Tally? Tally, I have a kind of previous comment, but I think you said that towards the end. Part of the limitation with the genotyping platform we have today is because they've been designed on wheat, like with that limitation on the D diversity. That's and what you're saying we're going to build now this next generation that we will. This is based on what? On genotyping synthetics, or going back to Taoshai and combining diversity from Taoshai into the SNP chip. So what's the?
I think the new genotyping array that Bristol have brought out, I think it's meant to have wider diversity to pick the variants to include in the genotyping array. I did have a quick look recently, and it looks like they used a lot of Watkins lines in there. So I'm not sure how far the diversity goes back. You know, CIMMYT have also been involved as well, so maybe they have looked at their dgenome GPOL. It'll be interesting to see how better the enrichment is across the dgenome compared to the current arrays. I mean, array genotyping is easy because you can send it away, get it back, and it's fairly easy to then process the data quickly if you want. Sequencing is probably the next step, and with that we just need to improve our capability of processing that data, I think, for the research, because that's how we'll get the ultimate resolution across the dgenomes that we need.
My only question, which is related to that, which I think you answered very at the end, is how much once you find this novelty in the synthetics, you go back to the true D, the Taoshai genome. Do you really see them? I mean, clearly you need to see those alleles because they will be coming from D, and probably how much we could do potentially in Taoshai directly to like direct work.
That's a really good question. It's something that we've been thinking about a lot at the moment. So we currently have Wright here, who's an MPhil student, and he's working on an Age-loss Taoshai population, so treating the original Age-loss Taoshai collection as an association mapping panel. And that might be the answer to the power questions that I've been thinking about, because if we find a QTL in that association mapping panel, will find something interesting, we then track that through the development of the synthetic. So how does it change when you get to primary synthetic? How does it change when you back cross it to a UK adapted background? And I think it's a really nice study because you've got a diploid, a tetraploid, and then the final hexaploid. So there's a lot of stuff that can happen from a Taoshai to that final stage. So it might be suppressed, and that's the sort of thing we're interested. So I think thinking about it that way, going back to the resource, it is something we are more focused on. The problem is that we haven't grown the seed for so long, so that's what we're going through at the moment, is trying to get every line back germinated to make sure that the resource that we have keeps going forward. But I think it's good to think about it from that angle, definitely.
And Tally, maybe a last point I wanted to make. Oh, sorry, there's a question from Richard.
So I was thinking of those gene expression studies where they identify subsets of genes which are only expressed on one of the three genomes. I wonder whether, with your new synthetic genome weeks, you'd be interested to do that again to see whether you get other genes coming up as being uniquely gene.
Yeah, exactly. I think it's the perfect. I think synthetics are the perfect model for that sort of experiment. And I guess if we find a QTL like we have done for yellow rust resistance, we can then see how it's expressed across genome. Yeah.
Good time. I can
Yeah, please do this.
Charlie, I mean, you probably I've mentioned this, but when you've looked at the synthetics that are selected by breeders, how discerning are they? What are all selecting the same stuff?
Yeah, that's a good point, because every breeder would have gone in with a different selection strategy, I imagine. And in some cases, I guess I've been looking at the overall picture, but there are certain genotypes in some nested populations where every breeder selected it. So in that case, I would imagine that it was more, that was kind of obvious choice. Where in some cases, things got a little bit wilder, and I guess it really comes down to breeder's preference in terms of selecting a bit more of the wacky stuff. Depends on the breeder, I suppose.
And I'm trying to remember, but is there a common maternal parent for synthetics, or the cytoplasmic differences in the family generally?
We typically made all the crosses the same way round. So the synthetic donor was always the pollinator, I think. Is that right, Phil? Or have I got
The
the wrong one?
synthetic itself, the tetraploid was always the female. Then when we made the F1 onto a cultivated
There's a
variety,
common cytoplasmic donor coming from the female lineage.
yeah,
For every one of the
Yeah.
noun populations.
yeah, we always use the synthetic as male. Yeah.
Thank you.
Thank you. Thank you, Tally.
Thanks, guys.
The next speaker is Professor Richard Mott from UCL. Thank you, Richard, for coming today.
Yeah, I think you.
Yeah. Yes, fantastic. If you want to share, just go on. That should come.
Yeah.
Yeah.
Great. Well, thank you very much for asking me. I'm on. Great pleasure. So, it's only a photo I have of Ian. This is for me. He's sitting over there somewhere. At UCL, or rather in probably Gordon Square, which is one of the nice bits of Bloomsbury. I have very fond memories of Ian. He was always fantastic to ask questions on. I was always bombarding him with emails saying, "Is this hype?" To which he would normally reply, "Yes." But anyway, I'll press on. So, Ian's, I think his big contribution was the idea of these multiparental populations. I won't dwell on all the aspects because I think everyone in this room knows what they are. But what I want to talk about is how we can use not only multiparentals but other things to try to bring things together. I apologise. I pulled this talk together from other talks, so there are some inconsistencies. So one of the problems we face is detecting QTLs in these large populations. And that really means that we need to make the population size as large as possible and to make the variance explained by the locus of interest as large as possible. So we can control the sample size if we can bring different diverse data sets together. We don't have quite so much control over allele frequency. We have really basically no control over the quantity beta, which is the phenotypic effect of a causal variant. So we need ways to combine data sets to increase our sample size and power, and to introduce as much double variation as we can. So the rules we use at the moment for sharing genetic data, and I'm thinking here more of human data, but the same thing really applies, I think, in the crop and animal breeding world is either you just share data and you just rely on you know, kindness of strangers not to abuse that trust, that sort of scalp honour. The advantage of that is you get the data in its entirety, and you know, you can do analyses that you hadn't even considered when you first started. Okay. Then the opposite extreme, this is a trust no one approach. If you go and talk to any computer scientist who works in you know, data security, they're basically totally paranoid. You know, they will assume that at any point somebody is snooping, you know, and cycling off your data. But you know, there are methods from cryptography which are provably secure. I say provably secure, as what I mean by that is they are as secure as it's possible to be until we can factorize prime numbers. Rather, we can find prime factors of large numbers very quickly. Basically, most encryption methods rely on that problem being difficult. The trouble with these sort of very robust approaches is that the factor analyses that you can do are quite limited, and they're also quite slow. So the method I'm going to describe here is something between these two. Okay, it's not provably secure, but it's certainly a lot better than just distributing your raw data and hoping that people behave honourably. So, if we don't know or reveal an individual's genome, how can we find association between genetic variants and phenotypes? And how can we share genotypes and phenotypes across studies to improve power? So where it says human, you have to cross that out and say wheat. Okay. So I mean, obviously this is important because we do have potentially very large resources available. But these resources are often locked up. Either in the case of plant breeding companies, in the case of human genetics, they're locked behind various biobank laws, or companies like 23andMe. Although I think 23andMe have been hacked, so where are we? Yeah, so we want to be able to combine data across studies. And we clearly need ways to accelerate crop improvement to take account of the challenges that we face. So this is sometimes called homomorphic encryption. So the homomorphic means we want to encrypt our data in such a way that it's structure preserving, that we can still perform certain types of analysis as if we hadn't encrypted. Okay. Well, that's the key idea. So just to set the scene, this is meant to represent a matrix of genotypes. So these are genotype dosages, there's color coding. Okay, and this is for heterozygous individuals. Sometimes a genotype 0, sometimes it's 1, 2. And here we have a vector of phenotypes, which will be probably continuously valued. So the things that we want to keep private about this data are the rows, that's individuals. And the things that we want to make public are those columns, right? We want to be able to determine what's the allelic effect of a SNP on a trait, but we don't want to reveal anything about the rows. So, way to think about this is geometrically. What you have to imagine is you've got a hypersphere in a very, very high dimensional space. Okay, and dimension will be the number of individuals in your data set. And every individual is a point on a unit sphere in this high dimensional space. Okay. Sorry, not every individual. Every SNP, every genetic marker is a point on the surface. And every phenotype is also a point on the surface. Okay. Then it's not hard to show that what you really care about are the angles between these SNPs and the angles between the phenotype and SNPs. Because that angle is essentially well, the cosine of that angle is basically the correlation coefficient between a pair of the phenotype and the SNP, or the R squared, the R value of the Pearson correlation. So if you know the angles, you know everything. Okay, you can calculate p-values once you know the angles. So you've got this hypersphere, and therefore it doesn't really matter what your coordinate axes are. Okay, you can imagine spinning your coordinates, and the essential nature of that sphere stays the same. The relative positions of the SNPs on this sphere are unchanged, but your coordinate system makes them appear to be different. I should say that when we do, say, a mixed model to allow for unequal relations between individuals, what we're doing is we're basically squishing this hypersphere in some directions and stretching it in others. Okay. So I think this geometrical interpretation is quite nice. So how do you change the coordinate system? Well, mathematically, that means you need to construct what's called an orthogonal matrix. Okay, and the dimension of that matrix is going to be the dimension of this hypersphere. And when you multiply that matrix into the matrix of genotypes, all you're doing is you're kind of spinning the coordinates around. Okay, it doesn't change any of the fundamental relationships between the SNPs and the SNPs and the phenotype. And the key thing about orthogonal matrices is that this lovely property, transpose of the matrix on itself is the identity. So they are kind of self-inverse. And why do you care about that? Well, because we normally assume that all our data are normally distributed. So the likelihood of our data is the Gaussian likelihood, and the Gaussian likelihood is invariant under orthogonal transformation. Okay, so the likelihood you'll get if you apply one of these orthogonal transformations is exactly the same. That's where the homomorphic side of things comes from. So essentially, what homomorphic encryption would mean is that rather than doing the analysis along the top in black, which is like the plain text analysis, you would transform your phenotypes and your genotypes by multiplying them by this orthogonal matrix. And it would also turn out that your variance matrix would get transformed in a physical way as well. So you're now in ciphertext space. You can compute your likelihood. It's going to end up being exactly the same as the plain text. And then you go back here. So basically you get exactly the same parameter estimates. You get the same estimates of heritability. And so you can essentially do the same quantitative genetics. So formally, the way we think about this is our data set is some triple of things. It's our phenotypes, our covariates, and our genotypes. So we just think of them as a collection of vectors and matrices. Okay, and we want to find transformations, like they're called phi, which take one of these data sets to another data set. Okay, so phi obscures the data of the individuals, but it maintains the relationships. It obscures both the phenotypes of the individuals and the relationships between individuals, but it preserves relationships between SNPs, and you can still use things like a mixed model, okay, to get estimates of heritability and so forth. And crucially, the likelihood of the data is the same. And this also has the property, as I showed above, that if you want to combine data from different studies or different groups, you can do that. Okay, so in addition to doing a standard mixed model, people these days do various Bayesian alphabet type detail analyses, and these basically correspond to choosing different prior distributions on the SNP effect. Okay, so standard mixed model is the same as cheap luck, more or less, with regression. Well, if you choose a hyperparameter appropriate, and that very corresponds to assuming that the prior distributions of all the snippet vectors follows some Gaussian. But you might want to assume it follows the double exponential when you have a lasso model, or maybe you've got an atom at 0, so you've got a high probability that the effect was simply 0, then you've got some kind of inverse chi-square, or you may have some other explanation. The point is that your posterior distribution on your simple effects is going to be the Gaussian likelihood multiplied by this prior. And the prior, of course, doesn't depend on the data. So if you apply one of these orthogonal transformations, because the likelihood remains the same, okay, on the transformation, the posterior distribution for the encrypted data will be the same as the posterior distribution for the unencrypted data. So let's see how this works in practice. So on the left-hand side, I have some unencrypted genotypes, and then we apply some random rotation. And this is what the encrypted data are going to look like. And what is interesting is that, okay, if you take any one of these columns, you're going to get basically a trimodal distribution of genotype residues. Okay. It would be, if the data were perfect, you'd have quite exactly here. These are, I think, imputed genotypes, so the dosages are not exactly perfect. But after rotation, the distribution of these values is basically accounted. So what this means is the encrypted genotypes no longer look like genotypes at all, they just look like random numbers drawn from a Gaussian distribution. What you're actually doing is you're replacing the individuals by random combinations of each other. Okay, so in no sense can you talk about the in the encrypted space, is a row in the encrypted matrix no longer corresponds to one individual. It's some random superposition of all the individuals. So this is some data using rats. I'm just using standard mixed model and I'm calculating the genetic association of every SNP with the phenotype, which is platelet levels, what matter it is. And on the y-axis, we have the log p that we would get in the unencrypted plain text. And on the x-axis, we have the log p that we get with the encrypted ciphertext. And you can see that the numbers are virtually identical. If you calculate the heritability for this trait, it's also very similar. So you get a very small differences due to rounding errors, but basically mixed models work identically under these two conditions. This is from work done by Al Chen and colleagues with UC Davis. So they were interested in making sure that these Bayesian alphabet models actually do work. Okay, but they, although theoretically they ought to work, they rely on MCMC and all sorts of sophisticated iterative processes, and it's possible that when you could move from having genotypes which are integers to having genotypes which are sort of all over the place, perhaps these methods wouldn't work so well. But in fact, they do. So this is looking at breeding values. So using the Bayes CEI model, I can't remember which one it is. Essentially the correlation between the raw and encrypted breeding values goes through. It's also the case that there's no correlation that's observable between the original and the encrypted genotypes. So what about sharing data, or federating data together? So supposing you have three different studies, so these might be data sets from three different companies or just different universities. The idea is that each group will randomly transform their own data. They won't share what that transformation matrix was. All they do is they share the rotated data. So I'll put cubes on their sides to indicate the rotation. And then you want to combine those three rotated data sets and then get the same quantitative genetics. And also to do that, you do have to have some prior coordination. Okay, you need to make sure that every study is going to use the same set of sets. Okay, the same type of covariates. You've got to get rid of missing data. There's some bookkeeping kind of stuff that has to be done ahead of time. But once you've done that, each group can independently rotate their data and share it. So the way it works is the following. So each group will create their own private rotation matrix. It's called P1, P2, P3. And it's as if you stack all these different data sets on top of each other. But the encrypted data looks as if you had multiplied all the plain texts together, but with this matrix, which is a sort of block diagonal version of the individual encryption matrices. This is still an orthogonal matrix. Okay, so you've got a sort of large federated thing which is still made up of encrypted pieces. So you can do exactly the same analysis on this federated data set that you could on the original. Okay, this approach, it basically gives you identical results to most quantitative plain text genetics, because that's basically mixed models based in alphabet. You do have to assume a Gaussian likelihood. Okay, if you don't have a Gaussian likelihood, the method doesn't work, so it won't work for dichotomous traits, for example. But otherwise, it's basically a drop-in replacement. Okay, you don't have to change anything. Most of the outputs that you care about are the things like the snip effects, okay, and heritability. So those are unchanged by this whole procedure. So you don't have to decrypt those. Okay, so you can share these snip effects amongst all the groups. Okay. The additional computational complexity of this is not actually that bad. It's basically the order of n cubed, n is the number of individuals. Essentially, the extra computation comes from simulating a very large orthogonal matrix and then multiplying that into the genotypes. So it's certainly more secure than just distributing plain text, but we haven't proven yet that it's as secure as the slower manner. And there are, we are working at the moment on whether there are ways to improve this. So this is all in 2 papers that we published. Well, the second one just come out. So the first one kind of sets up the basic ideas, and this one uses these methods to, well, it applies to the basic alphabet, basically. So I think that's everything except for the obligatory acknowledgements. Any questions?
Thank you. Any questions? James.
Everyone's probably asked you, how difficult is it then, do you think, to crack it in terms of really difficult, really like multiples of really? Is it just incredibly, impractically speaking, it's not practical?
Well, no one's got no one's going to come up with a method. It doesn't find this. We did actually put out a challenge. So the way the computer science community works is you put out a data set on the web and you say there's $1,000 available if you can crack it. And I think it was the rat data we put up. And nobody did crack it.
Maybe you need to put more than 1,000.
I could write a grant where the subplots of the grant is put a million pound bounty on this.
And there was a second part, and then so assuming it's really difficult to crack, so have you managed to find groups of companies or institutes or whatever which in practice would scale in the way that you described the combined data set?
Not yet. We're having a bit more success with the animal breeding community through our association with UC Davis. One of my reasons for talking here is I'm trying to drum up interest in the farm breeding community here. I don't really know to what extent there are large data sets sitting in behind closed doors that could usefully.
No, there will be. For me, my view is that. Sorry, James.
I was going to say, I mean, you could imagine. I mean, there's readers here. The trials they run every year internally. I mean, it's just rich, and if they're able then, because normally the way it would work is, you give it to someone, perhaps like Niab, we combine the data and then give the results back out. So this way, each breeding company can get the benefits of the full data set themselves and do more messing around and explore the data more fully. So yeah, I mean, there's potentially lots of data.
Yeah, I know kind of ways you can think about doing this, and you need to have like a trusted broker, which you might have. That's all the data and does the analysis, or each group,
But Richard, for what you said at the end, bro, you don't even need that because you can create your own encryption and push this.
Yeah.
That I understood correctly. Yeah. You can create that.
Yeah, so you could. I mean, if you're completely trusted, you could just encrypt your data and stick it on the website.
Okay. And then people can combine with that final construction.
Yeah.
One question I have in terms of, I assume here that there is an independent, the way you see SNPs are as an independent variable, or random variable. Do we miss any information that could be linked to haplotypes, or am I missing something here, or you are assuming, because that information is potentially, well, not necessarily gone, not really. Because what you're trying to do is clearly here is anonymize the individual, right? That's what you do. Yeah. Absolutely. And it's so difficult to reconstruct those individuals that you are certain that those individuals are, right? Coming back to James' question, it's going to be very hard to crack. I mean, you need to try millions of potential combinations, and you don't even know which ones are correct anyway. And then but in terms of haplotype information, I think you are preserving that, right? There's no connection between SNPs. That's what I mean.
You preserve the LD between SNPs.
Yeah. Well, I'm happy that I asked a difficult question then.
I think, okay, well, one way to do it is you can think of, if you have a haplotype, let's suppose you think there are a maximum of 8 haplotypes segregating at each locus, for example. Then you kind of need 7 numbers to describe the relative proportion. So you could create a genotype matrix, which basically, instead of having a single column for each SNP, has 7 columns.
Yes, sir. Okay.
Okay, and then I think
you almost consider the haplotype like a genotype itself, like a point in your.
Yeah. I think you'd have to do a bit of pre-processing, and I'd need to think through whether there were some things which make it go wrong. But I think that would still work.
The computational cost is a bit of a problem, but you amortize this with time because you do it only once, if I understand correctly.
Yeah, well, the computational cost is basically no worse than the cost of doing the mixed model anyway. Okay. It's of the order of cubing the number of individuals. And it takes, if you want to make, say, a random orthogonal matrix with 10,000 by 10,000, that takes a few hours on a server, it's maybe 2 hours. It's not kind of months.
Good. Well, thank you, Richard, for this very interesting talk, and thanks everyone for the presentation. A round of applause for everyone. the intention of the rest of the afternoon is very much have a reflection of the day today. I would like to first of all say thank you to everyone that presented today and gave such a good account of the work that Mac has done over the years in collaboration with you. But one point I wanted to stress is this spirit of collaboration, which is something which clearly defines science. And although all of you have stressed that point, I thought that coming to the end of the event today is something which I wanted to rescue from my end, is because there is now a responsibility for us to take that legacy, and I would like to take this as a challenge for us. Clearly, we had the privilege to work with Matt, someone who could see things much faster than anyone else, and I think it's something which I agree with you. Sometimes coming out of having a conversation in the office, I think something Haidee mentioned. Yeah, you end up, you thought that you understood, you were out and said, what was that? And then, but that was something that you could always come back and get a second round. And that for me was always something that I take with me as more than just, if you like, the science, a lesson in terms of how we should conduct science, right, which is collaborative in nature. And I think it's something which has been stressed today. For me, that's something which I wanted to kind of share with you, because something which was very strongly coming from the different presentations, different topics. There's a common theme across all these topics, but for me, one strength is working together. We can do more than just working independently. It's something which clearly Niab would like to develop further. I'm sure Wayne, you will agree with me. But Wayne, if you wanted to follow with some other reflections.
Well, just to echo those points, Mario, and just to say that I think today has been a great opportunity to share and give us some sort of insights into Mark's legacy, and we miss him, but also his legacy will live on, and I think it will live on not only in terms of the scientific outputs, but in terms of the people, and we see that here. I think his legacy is in good hands. I just wondered whether the family, June, Alexandra, whether you want to offer any comments and to round up with.
Just to say thank you to everyone for sort of hosting this and having us here today. I think when Wayne invited us, I said that one of my reflections is that I don't really know much about what Dad did. And we've got a bit of an insight into that today. Get lost a few times, but tried to keep up as best I could. So yeah, just a big thank you for us for putting this on and having us here.
Thank you.
Thank you.
So off the agenda, but John Hickey and I are meeting at 5 o'clock in the Eagle for beer. Anyone who wants to join us, and we raise a glass to Mark, you're most welcome. 5 o'clock in the Eagle. Otherwise, I think it's tea, right?
Thank you, of course I will. Yeah. Thank you very much. Thank you everyone for coming today, and thanks everyone that as well connected from remote places. A great legacy of the work that Mac has done over so many years and with so many of you.
Could I just offer a word of thanks as well? So Mike Smith isn't here, but he works with me on Niab, and a big thank you to Mike for the hard work, and to Ross as well.
Indeed, yes, the team here as well. Thank you. Thank you everyone. Thank you. Cheers.